think so. Okay, so Kwon, thanks so much for invites. It's my pleasure to present result on behalf of our International Hydro-seq Consortium. And as you can see, this paper just published last year in November in Nature Medicine. It's a it's a mega paper, so I'm not going to tell you all the data, but I will give you an overview of what we have done for this paper. Okay, so just bear with me. So here's some introduction for hydrocephalus. Hydrocephalus has been classically defined as a enlargement of cerebral ventricle result from an inadequate passage of cerebral spinal fluid from its point of production within the cerebral ventricle to its point of absorption into the system circulation. So it is estimated that congenital hydrocephalus affect about 1 in 1,000 live births. And here is a picture of the brain in normal baby versus baby with the hydrocephalus. And you can see the normal ventricle look like this, and in patient with hydrocephalus, there's a large ventricle. And CF shunting has been the primary therapy for hydrocephalus with very high rates of failure and morbidity. And the population-based study has estimated that about 40% of all cases are predicted to have a genetic etiology. However, the causes of most of the hydrocephalus patients remain unknown. And there is a hypothesis that hydrocephalus is just driven by imbalance between CSF production and reabsorption, which means it's a dreaming issue. However, we challenge this hypothesis in this paper, and then hopefully after you see our data, you'll see there is a turn hypothesis for hydrocephalus causality. So here is just a brief overview of our study design. So our goal is to identify novel genes associated with hydrocephalus and its mechanism. So we perform whole exome sequencing combined with deep phenotyping and multi-omics data in 381 patients of unknown genetic etiology. And then we conduct a genomic and statistical analysis to identify genes enriched for de novo and rare transmitted mutations. And then we use bulk and single-cell RNA-seq data of human and mouse prenatal brains to characterize the spatial temporal dynamics of known high confidence and probable CH genes. And we finally show that with additional analysis that include new cases, more precise genotype phenotype characterization, and multi-omics data, this in combination could endow individually optimal treatment regimens. So here are the demographic characterization of 381 patients studied in this cohort. About 52% are female and 80% are Europeans. Interestingly, I want to indicate about 9% of all cases are consanguineous versus 1.5% in the autism sibling control cohort from Simon Simplex. And I will show you why this is important for our results later. So we first look for mutation in known CHG genes. We found 8 probands with rare damaging mutation in these genes, accounting for about 2% of our cases. 4 male proband have rare hemizygous mutation in L1CAM, which is the cause of X-linked hydrocephalus and aqueductal stenosis. Proband demonstrate a typical feature of development delay, microcephaly, adducted thumbs, and paralysis. Another CH patient carried novel compound head in MPDZ, which is the cause of autosomal recessive non-syndromic hydrocephalus type 2. And 2 male patients had rare maternally transmitted DNMs mutation in FNN, the cause of X-linked dominant periventricle heterotopia, whereas their mothers were not on the phenotypic spectrum of the disease. And 1 male patient harbored a compound head in CRB2, which is a cause of autosomal recessive ventriculomegaly with cystic kidney disease. Overall, we estimate, again, the known CH gene can account for 2% of the sporadic CH, although we believe all of these 381 patients have been screened for known genetic cause for hydrocephalus. So that means deep sequencing, comprehensive characterization of genetic architecture, or at least the patient, again, or data reanalysis is important because you can always find a new mutation that was previously missed. So for the downstream analysis, we remove this patient from the analysis, resulting in 373 proband, including 225 trios. So next, we test different functional groups of variants in cases on the left and control on the right. And we perform this de novo analysis on either all genes or genes that are intolerant to loss of function mutation based on NOMAD. And show on the number of observed de novo mutation, expected de novo mutation, de novo enrichment, and p-value. And you can see damaging mutation are significant enriched in all genes, and with with a stronger enrichment when we test the de novo mutation in genes that are intolerant to loss of function mutation. Therefore, we estimate that damaging de novo mutation can account for about 18% of the cases. So in this cohort, there are 12 genes with 2 or more de novo mutation, as compared to 2.7 based on our simulation. And the enrichment of de novo mutation in 5 genes reach genome-wide state. You can see TRIM71, PTEN, and SMARCC1, FOXJ1, PIK3CA. There are 3 other loss of function intolerant genes exhibit at least 2 protein altering de novo mutation, and they are MTOR, PASH1, forming 2. There are 4 other genes with PLI equal to 0, each having at least 2 protein altering de novo mutation. So in our previous paper published in Neuron 2018, we identified significant enrichment in TRIM71, which is an E3 ubiquitin ligase, and SMARCC1, which is a member of BAF complex. In this expanded cohort, we identified 3 new damaging de novo mutation in TRIM71 and 1 more in SMARCC1. So in total, TRIM71 now harbors 6 de novo mutation, including 3 R603H and 3 R796H mutation. They map respectively to homologous position in the extremely conserved 1st and 5th place of TRIM71 RNA binding NHL domain. We also found previously unidentified unfaced damaging mutation N701K localized to the 3rd place of the NHL domain and predict to destabilize protein RNA interaction. And I want to say the strong clustering of TRIM71 mutation in initial domain suggests that this mutation might lead to selective loss of particular TRIM2 function. And SMARCC1 now harbors 3 de novo mutation, 2 transmitted loss of function mutation, 1 unfaced loss of function mutation, and 1 transmitted demis mutation. We want to point out 5 of these 7 SMARCC1 mutation are likely loss of function. And our data are consistent with haploinsufficiency as a primary mechanism by which SMARCC1 increase the disease risk. So we also observe so this is very relevant to what Bobo just showed. So we also observe significant enrichment of mutation burden in PIK3 signaling pathway. So PIK3 encodes the PIK kinase P110 alpha subunit that phosphorylate the PIP2, okay, and then to produce the PIP3. You can see my cursor, hopefully. And then PIP3 binds and stimulates the AKT kinase, leading to mTOR activation. And the PTEN encodes the PIP3 phosphatase that reverse this reaction. I want to say this PIK3 pathway gene have been shown to regulate the growth, proliferation, and differentiation of cells in multiple tissues, including new stem cells in the development ventricle zone. A related germline mosaic mutation has been defined in multiple brain and body overgrowth syndromes. I want to point out in our cohort, these are not somatic mutation. These are germline mutation with a little balancing between 43 and 53%. So we know PIK3, you can see that harbor 3 de novo mutation in unrelated patient with shunt hydrocephalus, microcephaly, meganecephaly, polymicrogyria, and craniofacial abnormalities. And then I want to say this all 3 de novo mutation have been linked to meganecephaly capillary malformation, polymicrogyria syndrome, and all localized to a recurrent mutation in cancer. However, I want to say that although, because I don't have too much time to talk about all the detail, I want to say, although all of our PIK3 patients retrospectively met the clinical criteria for the NCAP syndrome, none of them carry a diagnosis before involvement into our studies. And then PTEN also contain multiple de novo mutation, and then some of them are cancer hotspots. I also want to say, PTEN mutation are known to cause PTEN hematoma tumor syndrome. And again, when we screen this patient in the clinic, none of them have this this tumor syndrome. And MTOR also harbor, multiple de novo mutation, multiple transmitted unphased rare mutation. And again, this patient have not met Smith-Kingsmore syndrome, which are known to be associated with mTOR germline mutation. So this observation suggests distinct feature of no-Mendelian syndrome in sporadic patient can be overlooked by clinical caregiver, providing significant and important consideration of genetic screening of newly diagnosed congenital hydrocephalus children. Okay, so we also found the FOXJ1 with 2 or more de novo mutation and 1 transmitted DNMs mutation. I want to say that all the FOXJ1 patients exhibit obstructive hydrocephalus with aqueductal stenosis, and foaming as well, 2 de novo mutation. This patient have obstructive hydrocephalus and aqueductal stenosis. And PATH1 is a gene we previously identified in neuron paper. And here, I just want to point out, we only identified 3 patients in previous paper, and now we have more PATH1 mutation, providing additional evidence suggesting that PATH1 is a bona fide hydrocephalus gene. So we went over the de novo analysis. Now we want to exhaust our analysis for transmitted mutation field. So we perform burden analysis and identify another gene called FXYD2, which is involving the calcium signaling. And then you can see one of these mutation is a recurrent one, okay? And then our bioinformatics analysis suggests that this mutation might cause exon skipping, providing additional evidence for FXYD2 is a bona fide, hydrocephalus gene. However, I want to say in vivo and in vitro study need to be conducted to confirm the pathogenesis and figure out the mechanism. So now it's really important because, as I point out, there are about 9% of our congenital hydrocephalus patients being consanguineous. So this suggests there might be enrichment of recessive gene in our cohort. And indeed, I just want to quickly go over the result because we probably will run out of time. There are significant enrichment of recessive genotype in genes known to cause hydrocephalus in mouse. And then this is another syndrome, okay, dystrophloglycanopathy, and with hydrocephalus as one of the phenotype. And you can see when we test 12 genes that are known to cause this syndrome, there is a significant enrichment. I also want to point out, in the mouse screen, most of the genes are completely knocked out, right? So it's a recessive model. So among this patient, we identified six patients carry recessive genotype in mouse hydrocephalus genes. So in this slide, when we stratify patients based on whether they are consanguineous or they are not consanguineous, you can see 5 out of 6 recessive genotype are clustering in consanguineous cases. Therefore, it demonstrates that consanguinity is a big, predictor, or is a big factor for congenital hydrocephalus. And we estimate that 15% of consanguineous cases result from rare recessive genotype in the mouse congenital hydrocephalus genes. So next, we want to figure out some mechanisms, providing some mechanistic insights into the genes identified through the study. So we perform, and I just want to give you some heads up. Most of these genes are involved in neural stem cell fate pathway. So we test whether this high confident or probable gene converge in gene co-expression network and or cell types in the developing human brain using weighted gene co-expression network analysis developed by Dan Gashwine at UCLA. So we analyzed the bulk RNA transcriptional atlas of the mid-gestational human cortex to identify distinct module or network of genes. And interestingly, we see an enrichment of yellow module using this method in our hydrocephalus patient. I would say high confident and probable genes. And this yellow module also enriched for known for the DDD gene list, as well as some simplex autism gene list. And when we test when we use this yellow module genes, and then test what GO turn and HPO turn are enriched in this yellow module, you can see this yellow module are enriched for mRNA processing, RNA splicing, chromatin modification for the known GO turn, as well as some HPO turn that relate to craniofacial defect, as well as autism spectrum disorders. And next, we want to do more detailed analysis using single cell RNA-seq data to figure out whether there's any specific cell type marker that are enriched in our congenital hydrocephalus cohort. So indeed, you can see that when we analyze our high confident probable or known hydrocephalus genes, you can see that the cell marker for excitatory neuron, intermediate, and this cyclin progenitor S phase and cyclin progenitor G2 N phase are enriched in our hydrocephalus patient. In contrast, the developmental disorder and autism, they are enriched for other cell type, and these genes are expressed in the later stage of the brain development. So I just want to do a quick conclusion. So using our cohort by doing whole exome sequencing and deep phenotyping plus a multi-omic analysis, we can find the genetic cause for about 22% of our cases. And then this analysis further established TRIM71 and SMARCC1 as bona fide risk genes and identify new genes, including PIK53A, PTEN, mTOR, FOXJ1, F-formin2, PATH1F, and FXYD2. And we show a very important concept, which is distinct feature of known Mendelian syndrome in sporadic hydrocephalus program can be overlooked by doctors in a clinic, which suggests that we need to perform genetic screening using exome sequencing or whole genome sequencing of newly diagnosed hydrocephalus children. We also found overlap genetic risk factor between hydrocephalus and autism and other developmental disorder. And we also see a difference in the expression profiling, which means in our paper we show that hydrocephalus high confidence genes express earlier than those for autism spectrum and developmental disorders. And we further demonstrated that typically people think hydrocephalus is due to abnormal CSF dynamics. However, we show there's an alternative route, which means if you have a genes that disrupt your early brain development, like PTEN, MTOR, PIK3, TRIM71, or CDR pathway, FOXJ1, you might develop hydrocephalus. So with that, I want to thank my previous boss, Rick Lifton, and my commenter, Chris Colley, as well as my line member now at WashU. And I need to thank Dan Gashwine at UCLA for providing the RSC data and continued support for my current project and grant application. And here is my funding resource. And with that, happy to take any question.
Thank you, Peter, for presenting such a huge genetic studies. Anyone, again, feel free to open up and ask questions or through chat. I guess I'll start. So I don't understand trail sequencing field that well. Is the reason to use autism as a control because there's a lot of autism trails being sequenced? Yeah,
that's a great question. So you probably are aware in autism consortium, they sequence quads, which means they sequence affected proband, unaffected sibling plus parents. So in our, of course, we know there is overlap across multiple neurodevelopmental disorder. Although we use the unaffected sibling as a control, but it's not ideal. But I think through multiple study, we basically take a cheaper way to conduct study. We say, hey, if we can see de novo mutation in our cases, we test our unaffected sibling control. If we see no enrichment of this gene set, we think it's safe to say that, because we suspect there might be some impact of the genes, for our hydrocephalus gene on this autism spectrum, right? But if we could not see enrichment of de novo mutation in this unaffected sibling, I think we are safe to say, hey, this gene are specific for congenital hydrocephalus. Okay.
Yeah, makes sense.
I have a question.
Go ahead.
So PIK3CA is pretty often like somatic. Do you think that there are cases that you're missing? Not that you're missing, but that we're missing in general by testing blood?
That's a terrific question. I want to say we need to sequence deeper to understand whether it's truly germline or actually it could be parental mosaic, right? Yeah, so based on our current evidence, we cannot rule out the possibility that the de novo mutation we identify here are parental mosaic, okay? So I think this is a challenging question and a very good question to figure out. I'm not saying that these are definitely germline mutation, but based on our data, they are more likely to be, germline mutation. I hope this answers your question. And I want to point out, most of this sample are from saliva. We don't have blood because we use social media to recruit this patient. So, there are a lot of experiments we need to figure out, right? The best way to do this experiment is to sequence affected brain tissue, blood DNA, compare, right? To definitely answer this question. Okay?
Thanks.
Thanks.
So something along that line, because I really don't understand most of the germline variant analysis work. So you mentioned like, so this is like very basic questions. You mentioned like you can explain about 30% of the effect using using the known early novel mutations, right? And then so for the rest of those like say 70% of the cases, what do you think, like what are the reasons that you couldn't find any variants, and what's your approach or future direction?
Yeah, this is definitely a great question. So this is a power issue, in my opinion. We are underpowered, with only 225 patients. So I would assume when we reach the saturation point, and based on our simulation, we probably need close to 8,000 trios to reach saturation, right? Because we'll get more recurrent hit. So for generalized somatic analysis, one of the there are different hypotheses. So for our hypothesis here, we're testing monogenic. We were trying to find genes with large effect sufficient enough to cause a disease, right? So this is obvious a caveat, right? The hydrocephalus, like many other developmental disorders, they are not that simple. Monogenic cause can explain a bit, maybe 20 to 30%, but it could be very complex, right? Gene-gene interaction, gene-environment interaction. It could be, other unknown factor that we don't have sufficient information yet to figure out. So I would say larger sample size, more complex data set that you had for your brain tumor project, and more sophisticated statistical approach will pinpoint more novel genes.
That makes sense. Thank you.
Thanks. Any question?
Yeah, I guess while we wait for maybe one last question, I guess I want to ask another. So Peter, I guess, obviously, I think the direction there will always be trying to sequence more people. And maybe with tissue availability, there may be possibility of multi-omics.
Yep.
But in the meantime, is there other approach that you can probably, statistically mine existing data to get more signal?
Yeah,
Like,
that's a perfect question.
Yeah.
That's actually what we are doing now. There are so many different consortia, right, investigating hydrocephalus, right? So we're aggregating those cohorts to boost our power. From methodology point of view, we are also developing new method. For example, the slide you have seen here, they analyze one type of mutation at a time, right? There are several groups. They are working on joint analysis of different type of mutation. So one of the most famous examples is the TATA, right? Transmission and de novo mutation test, right? So they combine the summary statistics from transmitted mutation, de novo mutation, and more recently they try to combine the structural variation probability, right? So when you combine the information from different type of genetic variants, you have better power to pinpoint disease gene. And I also want to say, we are doing gene discovery for this paper. However, we need more study looking at mechanistics processes associated with the developmental disorder, right? So for hydrocephalus, there are existing mouse model and the Xenopus model and cell-based model that our collaborators are using to better characterize the mechanism. And I think there are a long way to go for all these human genetic studies, and exome sequencing definitely only scratch the surface, right? I would imagine the next step would be, hey, let's get the tissue, even though it's easier to say than done. Okay, get a tissue of the patient, try to figure out the importance of somatic mutation or parental mosaic, right, in all the brain disorder, right? Because you probably have seen a lot of study by Chris Bosch's lab. Mosaicism is ubiquitous in brain disorders, right? And then whole genome sequencing, right? Like 2 years ago, whole genome sequencing 2,000 plus. Now it's 800 per sample at WashU. So by doing whole genome sequencing, we can investigate the impact of non-coding variants, interaction of non-coding and coding variants for complex disease. Okay, so I just toss some potential direction and some action that we are taking in the field. Yeah, hopefully this answers your question.
Yeah, I think there's a lot of promising directions to go. So I think if there's not one last pressing question, I think Peter and Bobo both have strong internet profiles. People have further questions, feel free to, I guess, reach out to them and ask about their papers. But thanks everyone for participating, and thanks Bobo and Peter for the excellent presentations. We learned a lot.
Yeah, thanks, Kwon. Thanks, Bobo.
Thank you.
Bye.